Hi everyone and welcome to today's lecture on lipid structure and function. As always, I just want to remind you that there will be chalkboard note slides after each slide that I describe so that you can pause the video and jot down your notes. So when we talk about lipids, I just want to first point out the main roles because there are a lot of different types of lipids and they have three main roles. The first one is energy storage, then we have cell membranes, and then we have endocrine signaling. And we'll go into each of these different groups throughout today's lesson. The groups in terms of structure that I want to talk about, when you think of lipids, you will either find them in long open chain form, such as you see here with the fatty acids, where you end up having a polar head and a nonpolar tail. And then you also have fused ring lipids, okay? So for the first group, the open chain lipids, this will include fatty acids that you see here. It will also include triacylglycerols, sphingolipids, the phosphoacylglycerols, and glycolipids. And then for the fused ring lipids, that's going to be steroids. And we'll talk about the various forms of steroids throughout this lecture. So I want to go through each type of lipid one by one. The first big category that we have are the fatty acids. And because of the dietary health relation when it comes to fatty acids, I'm sure you've all heard a lot of the terminology we're going to go through today. When we talk about fatty acids, first of all, I want to point out that they have the carboxyl group, which is shown over here on each of these. And the carboxyl group is going to be the polar end. And then you have the long chain that is a hydrocarbon chain, and that's going to be the nonpolar tail. Now, the fact that you have a polar and nonpolar head end, that basically tells you that you are going to have a hydrophilic end and a hydrophobic end. Now, notice that the polar end is hydrophilic because if you think in terms of water, water is polar, like dissolves like, so any other polar molecules will be considered hydrophilic, okay? Now, when we look at these structures and you have a hydrophilic end and a hydrophobic end, that will tell you that it is amphipathic, okay? Amphipathic, it has both polar and nonpolar ends to it. Now, when we talk about fatty acids, you're going to see saturated, which are the ones that you always hear are bad for you. And then you have unsaturated, which are the healthier forms. Now, if there is a carbon-carbon double bond anywhere in that long hydrocarbon tail, then you will categorize that fatty acid as unsaturated. Okay, unsaturated because you can open up that bond and add some more hydrogens. Whereas this first one here is saturated because there are no double carbon-carbon bonds. Okay, so every bond is already being used up in that hydrocarbon tail. Okay, nothing for you to open up there. You'll also notice that we have the terms trans and cis, which only apply to unsaturated fatty acids and not saturated. We're going to talk about that in a moment. So now looking at those fatty acids a little bit more, I want to focus in on the difference between unsaturated and saturated. But before we go into those details, I want to draw your attention over here, okay? I want you to be able to read the coding of, how to, I'll say translate what kind of fatty acid you have based on when you see certain numbers written out, okay? So when you see a number, a colon symbol, and then another number. The first number tells you how many carbons are in that fatty acid. Okay, so here you see the number 18 first. That tells you that there are 18 carbons. After the colon, you then see the number 1. That tells you how many double bonds you have in that fatty acid. So since there's 1, you only see 1 double bond over here. Then you see a triangle with a number at the top of it, a superscript up there. Okay? That tells you the double bond location from this first, the carboxylic carbon over here. Okay, so since it's the number 9, that tells you you would count from here, and on the ninth carbon away from that initial carbon, the ninth carbon away from there, that's where that 1 double bond is found. Okay, so make sure you know this terminology or this coding. Now, sometimes if you have more than 1 double bond, you'll then see multiple numbers written after it. That way you can track exactly where the rest of the double bonds are. If instead of a 1 here, you saw a 0, that would tell you that there are no double bonds, which would tell you that that is a saturated fatty acid. Since you have a 1, if you see a 1 or greater number there, then you have an unsaturated fatty acid because it tells you that you have at least 1 double bond. Now, when you have just 1, that will be monounsaturated, like you see over here. Okay, 1 double bond. Okay, if it's polyunsaturated, that tells you that there would be more than 1 double bond. Now, when you have unsaturated fatty acids, because of that double bond, you can end up with the orientation being either trans or cis. And you'll notice the difference is that cis puts a kink in the arrangement of that fatty acid. Okay? And so when you have cis unsaturated, sometimes students can use the trick of thinking kinky sister. Okay, that cis has a kink to her. So you see that there is that kink or that curvature here. Okay. Now, trans instead will be linear and it will look just like saturated. Now, saturated, we do not use trans or cis to describe. Okay, trans and cis is only for unsaturated, and it's because of how that double bond is affecting the arrangement of that hydrocarbon chain. Now, when you look at unsaturated versus saturated fatty acids, I want you to note that the melting point varies depending on which you have. Okay, so degree of fatty acid saturation will alter the melting point. Unsaturated fatty acids, they are going to be liquid at room temperature because they have a lower melting point. Okay, whereas saturated fatty acids, because they have a higher melting point, okay, they are going to be solid at room temperature. Okay, so it has to be a higher temperature for them to melt into a liquid. Okay, so make sure you're clear on the melting point aspect. Also, I want to point out that trans unsaturated, so trans fats and saturated fats, because they are linear arrangement like this, you end up having them able to stack on top of them, on top of each other, much more. And so they can accumulate more and be solid, which is a major problem for health, okay? Because these two, saturated and trans fatty acids, because they're linear and stack on top of each other and accumulate, that's what can clog up your arteries, okay? And you'll notice on here, you tend to see that more with animal fats being saturated or being trans unsaturated, whereas vegetable fats, those tend to be more the healthier cis unsaturated. Now, sometimes you'll see unsaturated fatty acids labeled as omega fatty acids. They're associated with reduced cardiovascular disease, and so you'll see them labeled as heart healthy. and basically very good for you. We'll talk about in some other lectures why exactly that is that they're so beneficial to you. Now, what's interesting with the omega fatty acids is we just went over the terminology for how to identify the different parts of how a fatty acid is labeled. And so when we're talking about omega fatty acids, the most common one that you've probably heard of is omega 3. Sometimes you hear omega 6 or omega 9. The way that they are labeled is based on this end carbon here. So the terminal carbon, which is furthest away from the carboxylic end, this end carbon is known as the omega carbon. So whenever you want to number an omega fatty acid, for instance, if it's omega 3, that tells you that you count backwards from the omega carbon to the carbon where the first double bond is found. So in this case, this is an omega 3 fatty acid because you have the omega carbon here. So that would be 1, 2, 3. At the third carbon from the end, that's where the double bonds are first found. And it doesn't matter that there's more after that. So the other ones after that, you'll notice you have it labeled here that there are 3 total double bonds. But the omega is simply numbered based on the very first appearance of any double bond from the omega carbon. Okay, so again, when we look at this labeling, the first number would tell us that there are 18 carbons in total for that fatty acid. The 3 would tell you that it's unsaturated and that there are 3 double bonds. The triangle then tells you exactly where those double bonds are from the carboxylic carbon over here, that alpha carbon. So you have 1, 2, 3, 4, 5, 6, 7, 8, 9. So see how it's numbered in red? At 9, at 12, and at 15, away from the first carboxylic carbon over here, that's where you find those double bonds. So when numbering the double bonds in this terminology here, you go from carboxylic side. When figuring out the omega number, you go from the terminal end, from that omega side. Now, with unsaturated fatty acids, because there is at least one double bond, we can do what's called hydrogenation, and that's the commercial process of using hydrogen to reduce the double bond so that that fatty acid has now been saturated. Okay, so you see from here, it goes from having the double bond, and you notice since there's a kink in it, that would be cis, and then it becomes fully hydrogenated here, and you end up having it now saturated. So you notice that it went from 18,1 to 18,0. That tells you no double bonds because it is now saturated. Okay, now once that has been done and you have that saturated fatty acid now, that changes a few different things. First of all, remember, saturated raises the melting point. So whenever you think saturated, I want you to think that S of saturated, saturated solid, okay? So now that you have raised the melting point, it will stay solid at room temperature, okay? That is basically cheaper and it gives it a longer shelf life, so that's why they do this. But it also is a problem because it makes it more problematic for your health, basically. And they can either fully hydrogenate it or partially hydrogenate it. And when they partially hydrogenate it, then you end up with trans fats. So it'll still, be categorized based on based on that change. Now, it'll still be categorized as unhealthy because as you know, trans fats and saturated, we mentioned, both have a higher risk of cardiovascular disease. Because when you look at the picture of their arrangement, you can stack more of them on top of each other, and that ends up accumulating and clogging things like arteries. Now, fatty acids are the most common lipid that you hear about, but we also have waxes, and you'll notice with the waxes, you have a long chain fatty acid because you can see that carboxylic side would have been over here, but you have it linked to a long chain alcohol. So a whole lot of hydrocarbons, and then what would have been an OH group has now been linked to the long chain fatty acid. These guys here tend to have high melting points. So when something has a high melting point, what does that tell you about it? Well, think of a candle that's made of wax. It is solid at room temperature until you light a flame on it. But it's solid at room temperature because a high melting point. And a lot of times their main function will be either as a protective coat for plants or for animals. Okay, both end up using waxes as protective coats. Another form of lipids which are built off of fatty acids are the triacylglycerols. Tri because three, and you will notice that it is basically three fatty acids linked to glycerol. And the main function of triacylglycerols are for energy storage, but I want to point out an important thing about them right now. Since we said that it has three fatty acids, those fatty acids can either all be saturated, they can all be unsaturated, or you could have a mixture, let's say two saturated, one unsaturated, or two unsaturated, one saturated. Now, when you look at triacylglycerols in meat products, animal fats, they tend to contain more saturated fatty acids. So theirs would be more likely to either be all three of the fatty acids saturated or two out of the three are saturated. And again, the way you can tell from the coding here, from the numerical code, is that you see the zero next to the number of carbons. So that's two saturated and one unsaturated. Vegetable-based fats, so for instance, plant seed triacylglycerols, you notice they have more unsaturated fatty acids out of the three. So in this case, all three of the fatty acids of this triacylglycerol, when it's the plant seed, are unsaturated, okay? And specifically these three, because you see the number one, so one double bond, that tells you it's three monounsaturated fatty acids there. Okay, and so that's important because that means if there's more saturated fatty acids in the triacylglycerol, then this will more likely be solid at room temperature, whereas the plant-based ones will more likely be liquid at room temperature, and they are basically a healthier form of the fat. Now, fats are not just important to us or valuable to us in the food industry, but also they are important to us because of saponification. And what that means is a fancy way of saying soap making, okay? Because scientists always want things to sound fancy and cool. So, saponification simply means soap making. It's when they use ester hydrolysis of fat with lye and form soap. And what's interesting is how it actually works. If you picture, for instance, laundry, what happens is you have soap micelles in the water, and then when you agitate the soap, the soap micelles, the water, the fabric in laundry, the micelles break open, and then they surround or reform around the greasy dirt. And once they've formed around that, then they're able to remove the greasy dirt from the fabric. And so with those micelles now, you're removing the dirt as well. Other very important terminology when it comes to lipids are the lipoproteins, especially when it comes to your health and proper diet or lifestyle choices. And so the first term on this slide is chylomicrons. Another term you sometimes hear used for chylomicrons are ultra low density lipoproteins. And these are the largest of all lipoproteins. They're involved in transporting triglycerides. Now, whenever you see low density for lipoproteins, those are unhealthy. So if these are ultra low density lipoproteins, chylomicrons are going to be one of the most unhealthy of the lipoproteins. Okay. Now, ultimately, any of the lipoproteins will be responsible for the function of transporting dietary lipids and basically allowing cholesterol to move throughout the bloodstream or to be either stored or excreted, depending on which form of lipoprotein. When you see the two main categories that I'm sure all of you have heard before, which is HDL, so high density lipoproteins, versus LDL, which are low density lipoproteins, I want you to circle the H of HDL and remind yourself that H stands for healthy. Okay, so it actually stands for high density, but you can remind yourself H for healthy, L of LDL, think lousy. That's the bad one. So HDL is healthy because that's the good cholesterol that basically carries cholesterol back to your liver to be flushed out of your body. So it's for excretion of cholesterol. So you want high levels of HDL when you do your blood work. Low density, that's the lousy one. That's the bad cholesterol that will increase your risk of heart disease and strokes and plaques collecting throughout your body because LDL carries cholesterol that accumulates in the blood vessels as plaque, and they block off those blood vessels. Now, in order to get high HDL, you want to do things like eating the right foods, drinking things like green tea, having a lot of omega foods like salmon, exercising a lot, keeping your weight down, and having an active lifestyle without drinking alcohol, without smoking cigarettes, without doing drugs. If you are someone who smokes, drinks alcohol, lounges around a lot, not very active, you have excess weight, and you're eating a poor diet, then you're going to end up with high LDL, the bad one, and low HDL. Now, unfortunately, genetics also plays a factor, and sex as well. So, sex as in men and women, as you can see on this chart, not sex as in the activity, although we said active lifestyle does, good things for cholesterol. But that's a whole other lesson. So just keep in mind HDL is healthy. You want a lot of that. It's for excreting, getting rid of cholesterol. LDL is low, bad, lousy. Okay, it collects the cholesterol in your body. Now, at the beginning of this lecture, we mentioned that there are three main purposes for lipids. We said energy storage, cell membranes, and endocrine signaling. So here we start to look at cell membrane function, and there are three major types of lipids that you'll find in membranes. There are let me get the laser, the glycerol phospholipids, the sphingolipids, and cholesterol. The glycerol phospholipids, I want you to put a little star next to those, circle them as the most abundant of the membrane lipids. They're made up of two fatty acids attached to a phosphate and a glycerol. And then you have sphingolipids. For sphingolipids, I want you to immediately think of the nervous system. These are going to be most abundant in brain and the nervous system, and especially when you hear the term myelin sheath, which is around nerve cells, that's heavily made up of sphingolipids. And so when you think about defects or diseases that are going to affect sphingolipids, you're going to see severe deterioration of the nervous system, okay, brain and nerves. The last one is cholesterol, which is a steroid, and that will basically affect the fluidity of membranes. And it's also an important metabolic precursor that we'll talk about in later slides. Now, as I just mentioned a moment ago, when it comes to diseases of those sphingolipids, there are various diseases that can occur, but most of them you will see some sort of neurological issue because of the fact that sphingolipids are so significant in the myelin sheath and around nerves and in the brain. So the three that I have mentioned here that I want you to know are associated with sphingolipid metabolism defects. First, we have Tay-Sachs disease, and Tay-Sachs is basically a severe progressive deterioration of mental and physical abilities because there's been nerve damage. And ultimately, people with Tay-Sachs, they lose muscle control and will end up with problems like blindness, paralysis, and death. Then we have Fabry disease, which you don't hear as much. That one, you basically end up with pain in the hands and in the feet, and then you see skin rashes, especially in the extremities. The last one is Niemann-Pick syndrome. That one is going to basically cause elevated levels of sphingomyelin, and it causes an accumulation of lipids in cells and in various organs such as liver, spleen, brain, bone marrow, which can cause a cascade of various complications because you basically have lipids accumulating where they don't belong. And that then brings us to the third function, which is cell signaling. Uh, this question that I asked here is what significantly contributes to the cell signaling functions. And if you think back to other lectures that we've had where you think about what's actually involved in signaling and you need ligands binding to receptors, especially the pockets in receptor proteins, well, then you can see that hydrophobic property of lipids can contribute a lot to their ability to function in terms of having high affinity ligands bind to hydrophobic pockets in receptor proteins. So ultimately, I know that was a lot of, annoying sciencey words, but basically the key here is that the hydrophobic aspect of lipids is very valuable to their function as ligands that can then bind to hydrophobic receptors. And so all of the lipids that we've been basically focusing a lot on so far in this lecture have been the open chain, the long hydrocarbon chain based lipids. But we also mentioned that you have the fused ring lipids, which are the steroid hormones. And in terms of the different categories that you have, there are glucocorticoids, mineralocorticoids, androgens, and estrogens. And so the androgens are associated with masculine sex characteristics, whereas estrogens are associated more with feminized sexual characteristics. Now, ultimately, these are potent signaling molecules, and they'll be involved in things like cell development, reproductive biology, and organismal physiology. And we're going to go through some examples now. So, some of the really common steroid hormones that you hear the names of are progesterone, cortisol, aldosterone, testosterone, and estrogen. The first one, progesterone, this is very important in terms of menstruation and development of mammary tissues. So ultimately, importance in terms of breastfeeding and maternal aspects. Then you have cortisol, which you see it with liver metabolism and immune function. But the one I want you to remember most, especially as students, and especially as students in biochemistry, is cortisol is critical to your adaptation to stress. And you might have heard those commercials for a while that kept emphasizing how stress raises cortisol and then cortisol raises body fat. So, there's this whole pathway because they are lipids. You can end up having health complications from stress because it is messing with hormones, including cortisol. Next, we have aldosterone. This is very important in ion transport in the kidneys, but the big one that I want you to know for aldosterone is it's also very important in regulating your blood pressure. Okay, so again, progesterone, think menstruation and mammaries. Cortisol, think stress response. Aldosterone, think blood pressure. And then we have the sex hormones, which are testosterone for the development of male reproductive organs, and then estrogen for the development of female reproductive organs. And when you hear um, male hormones, think of the term androgens. That'll be important in a moment. So now a moment ago, I mentioned how I wanted you to remember the term androgens and how that related to masculine sex characteristics. So that's important when we talk about synthetic hormones because some of the examples of how we've modified chemical side groups on steroid hormones has given us valuable drug abilities when it comes to modifying biological activity. And so the two examples that I want you to be aware of and to recognize their names are nandrolone and bicalidamide. Okay, nandrolone and bicalidamide. So first we have nandrolone. This, as it says here, is a potent androgen agonist. And so when something is an agonist, that basically means that it can bind to the same receptor and trigger the same reaction or cascade that the intended ligand would do. So if the natural androgen promotes male reproductive development, then nandrolone as an agonist would also promote male reproductive development and male sexual characteristics. Okay, and so that's why you would see it used by bodybuilders to increase masculine secondary characteristics such as increased muscle mass. Then we have bicalidamide, and that one points out that instead of an agonist, it's an antagonist. And so androgen antagonists are going to bind and block the receptor that you're looking at. So in this case, it will block the male associated hormone, okay, that the androgen that would bind to that receptor. So over here, it shows that it's been used to treat prostate cancer, but another area that is also very valuable has been with secondary effect as promoting feminization in male to female transgender adolescents. And so conversion toward feminizing characteristics would be accomplished with bicalidamide because it's an androgen antagonist. Okay, so it blocks the masculine male associated androgen hormone from binding to the receptor. Then we get to vitamin D, and you may be asking yourself, well, how does that play into this? And I'm sorry, I start to laugh because that is one of my favorite memes. But when we think about how vitamin D is connected with our lesson so far, it's because as this sunshine vitamin, it's derived from cholesterol through UV light activation. Okay, and so in this figure here, you can see the UV light triggering the conversions that ultimately lead to the usable form of vitamin D. And just 10 minutes outdoors, although officially a lot of times they'll say 5 to 30 minutes outdoors, that can allow for the conversion necessary into the biologically active form of vitamin D that we need. The next category that we have, and this one will be the last of our categories of lipids, are the eicosanoids. Okay, very, very important. And with these, there are four main classes. There are the prostaglandins, the prostacyclins, thromboxanes, and the leukotrienes. And ultimately, these are involved in things that include inflammation. You see them in reproduction and gastric secretion and in regulating blood pressure. And they're derived from arachidonic acid. And with that, it's through what's called the COX cyclooxygenase enzymes. And since that is necessary, so if we look at this figure, since the COX enzyme is necessary for that conversion, then when you are looking to block prostaglandins from being produced, because for instance you don't want uncontrolled inflammation, then you would do that with COX inhibitors such as aspirin and other NSAIDs that are anti-inflammatory. And my brothers and their friends, the way that they always remembered that is they actually called one of the friends aspirin because they considered that friend a COX inhibitor, COX blocker. And I will let you do with that what you will in terms of helping you to remember that terminology. Uh, Now, the other one, so this is for prostaglandins because you see the cyclooxygenase enzymes on that pathway. The other one, leukotrienes, the way that those are inhibited is through inhalers. And so that will then, the inhaler will reduce the leukotrienes and bring down the kind of inflammation that would be causing difficulty breathing, for instance. Now, there are natural ways to block the formation of certain prostaglandins and of thromboxanes. And the main one is eating high omega foods like salmon. So, omega 3 fatty acids block prostaglandins and thromboxanes, and that in doing so, they also block or prevent clotting issues and prevent heart attack and strokes and issues like that. And like I said earlier, omegas are going to keep coming up because they are very healthy for you. So I do recommend you trying to get as much of the omega foods like salmon in your diet as possible because they really can help with a lot of different parts of your body, especially your cardiovascular system. And so that brings us to our review questions. Please make sure to send me the answers to the review questions in the Remind app, or if you're having trouble with the Remind app, let me know and you can contact me through email. There's one more review question here, and that is it for today. As always, if you have any questions or concerns, please contact me in the Remind app or through the email. Thank you and have a great day.